centrifugation. These types of centrifugation where the separation is based on only density. So the cell component separation is very difficult in the case of density gradient centrifugation because there is only a principle of density of a particle of a sample is going to be separated off in the case of density gradient centrifugation, where a particle density is going to be matched with the gradient density. Based on it, it is going to be separated off. Even a density gradient centrifugation is going to be classified as two types. A major important thing in the case of density gradient centrifugation is going to be gradient at different density. So a choosing of gradient is very important. So a gradient which is going to be good gradient, as we are going to see. A good gradient must have following characteristics. A gradient must not affect the bioactivity of a sample. This means a gradient used must not affect the sample. If we take a protein will undergo denaturation. So the gradient which will cause a denaturation will not be used based on the sample used. So a gradient should not be affect the bioactivity of a sample. Like that, a gradient should not affect the corrosion, because a corrosion of rota can also be possible if the gradient is corrosive. If it is corrosive, then accurate purity will not be possible. A gradient should not be sterilizable, and the gradient must be cheap. If it is in high cost, it's very uncomfortable for usage of a gradient for many times. So the gradient must be easily removable to form a purified sample. If it is difficult to remove, it may cause a decrease in purity. If the gradient will be attached with the sample and it's very difficult for the separation, it may decreases the purity. The gradient must not absorb the UV range, because a separated biomolecules will undergo further for colorimetry test and spectrometry test. If it will absorb the UV range, it may causes a deflection in the results of UV range. A deflection in the results of UV range will causes a difficulty in identification of component, because each biomolecules has a UV range, specific UV range. A protein has a different UV range and a carbohydrate has a different UV range. Like that, each one has a different UV range. A glucose has a different UV range. If a gradient is going to affect the UV range, it will cause a confusion whether it is a protein or a glucose. Like that, confusion will be possible. It must be decreased if the UV range absorbing gradient will not be used. A gradient must have increased water solubility, because it must be water soluble only can be used as a medium for the sample component. A common gradient can be used as sucrose, which is going to have a maximum density of 1.33, which means if we use a sucrose as a gradient, then biomolecules which are going to have a density of 1.33 can be separated in the sucrose gradient. If a medium is going to consist of sucrose, silica and cesium chloride, like that, a different medium is used. If a biomolecule is going to consist of 1.33, if it is a protein, then a 1.33 containing gradient, which is sucrose, is a region where the biomolecule protein is going to be separated off. Like that, there will be a standard density at which each medium is going to have, so that it can be useful for identifying the protein density, for identifying the biomolecule density. By identifying Identifying the biomolecule density, we can easily identify the biomolecule. So these are the standard gradients and its density. So some of the commonly used gradients are going to be discussed further. So in the case of sucrose, sucrose is going to be very common used gradient. So this gradient can also be used in differential centrifugation and density gradient centrifugation. It is commonly used because of its low cost and it's going to be very easy for the preparation of this compound. It is going to separate the component like Golgi apparatus, plasma membrane, smooth endoplasmic reticulum, mitochondria, lysosome, and the proteins. Soluble proteins can also be separated. Even a plant viruses can be separated off. An enterovirus can be separated off. Like that, different components can be separated because there is only a small differences in their density is possible. So, Golgi apparatus is going to be separated at 1.06, plasma membrane at 1.16. So, each density, based on its each density, is going to be separated off. A major disadvantage is going to be osmotic effect. Sucrose may causes osmotic effect. So, cells like viruses, by handling with the sucrose is very important to handle it perfectly. It may also penetrate the cell membrane and causes lysis of the cell. So, a usage of sucrose can also be causes the osmotic effect. A commonly used sucrose is 0.25 m, that is 0.25 molarity, which is very low concentration. Due to its low concentration, a microbial growth can also be possible. Due to the microbial growth, there will be a deflection in the sample quality. Next, a common used medium is going to be cesium chloride. It's going to have a density is 1.91 gram per centimeter cube. So it's going to be soluble in water at highest level. It's going to have a low viscosity. So due to its low viscosity, it is used commonly for RNA and DNA separation, where sucrose can't be used here. A major disadvantage is also here as osmotic effect. And it's going to have a high cost and absorption of light, which is not possible with other gradients. Then a phycol. A phycol is going to be a polymer of sucrose and epichlorohydrin. It is a non-ionic polymer and has a low osmotic effect. For the replacement of sucrose, a phycol came. A sedimentation is going to be 418 grams per centimeter cube. A major disadvantage is going to be highly viscous than sucrose, and it's also very costly than the sucrose. And it can be contamination possible because it is going to be a polymer of sucrose. So the contamination of microorganism is possible in the case of phycol. Then percol. A percol is going to be a polymer of pyrrolizone and coated colloidal silica. It's going to have a low viscosity for so DNA and RNA separation can also be possible. It's going to separate about 1 to 1.3 grams per centimeter cube of density. So these are the good medium or good gradients can be used of, which are going to be used as commonly. So now we are going to see about the density gradient centrifugation, how is going to be taken place. Even a density gradient centrifugation can be taken place before, as we already saw in differential centrifugation. We have saw as a first step is going to be sample preparation, and second thing is going to be gradient preparation. So here is also a sample preparation and gradient preparation is very important steps. As in the case of gradient preparation itself, there are about 2 steps possible. They are discontinuous and continuous process. But in the case of discontinuous process, where there is a gradient which has greater not going to be continuous. They are going to have a greater differences in their gradient density. That means a density of the gradient is going to be increases abruptly, where there is no steady changes or gradual changes. Gradual changes in density of a gradient is not possible in the case of discontinuous density gradient preparation. But in the case of continuous density gradient preparation, a density is going to be gradually changed, because in the preparation of discontinuous density gradient, one layer and another layer is going to have a greater differences in density, but there is going to be a density in the order of increasing order. So the layering of solution is going to decrease from top to below and going to increase from below to top. A layering of density gradient is going to be increased from top to below and going to be decreased from below to top, which means in the test tube a bottom gradient is going to have a higher density, and the density at the top is going to have a lower density, as a lower density component is going to be at the top and higher gradient is going to be at the bottom. Based on its density, these gradients are going to be layered off. The sample layering is going to be made at the top of the gradient, which has lower density. So these components, this density gradient, that is discontinuous density gradient, can be used in the subcellular organism fractionation. But a common method of density gradient centrifugation, a gradient layering, is continuous density gradient preparation, because it is going to be very accurate method for the separation of components. But it needs a more working, but it's going to be more accurate, where a discontinuous method, it's not more working, does not needs more manual, but a continuous is going to be more manual, and tedious work is needed for this method. In the case of continuous density gradient centrifugation, a density decreases from the bottom to the meniscus. It's also like discontinuous, where the highest density is at bottom and lowest density is at meniscus, that is at the top. But the method of gradient preparation is different. It is going to be as usual, where the gradient density differ more. But in the case of continuous density gradient preparation, a gradient is going to have a density of small differences only. This can be prepared off by this method. In this image, like this image, where there is a two test tubes, where there is a outlet. In this there will be a low dense solution present. In this there is going to be a high dense solution present, where there is a stirrer for continuous stirring of components. There is only one outlet. A test tube is there, where high density solution is on. A high density solution is going to be dripped into the test tube, then it was stopped, then the less dense solution is going to be poured from the less dense solution to the high dense solution, and it was stirred off. This component is again poured into the test tube, which is going to have high density solution. Like this, a small density different gradients can be possible by gradual introduction of low dense solution. Due to the gradual introduction of low dense solution, the gradient is going to be have a density of small differences is layered. So here is the explanation. It's going to have a two chambers. It is going to drip into the centrifuge tube as denser solution. A second, after that it will stop. Then a second chamber is going to have a less dense solution. A less dense solution is going to be poured into the first chamber. Then the level of a solution in first chamber is compensed by the second chamber. A stirring device is going to stir it. Based on this, different density solution can be possible. So thus the first chamber is going to have a less dense solution due to the introduction of less dense solution into our first chamber from the second chamber. So the solution dripped to the tube has decreasing density as a higher dense solution was poured at first, and the layering of lower dense solution can be poured off, which is going to be as gradual changes. Next we are going to, as we already saw, gradient preparation, we have saw how to prepare the gradient. Then we are going to see about the sample application. How is going to be a sample prepared and it was applied. A volume of sample is going to be applied across the cross section of the test tube, where there is about 2.5 centimeter test tube is used. Very lesser volume of a sample is going to be used in this method because a lesser volume of sample will have a higher resolution. A sample concentration is less as 10 times than the concentration of a starting gradient, because if the starting gradient has a higher density than the sample, then it may causes the sedimentation of the sample at faster rate. So a starting gradient concentration is going to be less as 10 times, high as 10 times than the sample. So for the gradual application of the sample, a syringe is used for this technique. A syringe is inclined at 45-degree angle for the application of a sample on the test tube cross section. For the DNA, there will be a more fragile method can be used. Instead of syringe, a mouth pipettes, a broad mouth pipettes can be used. So these gradient are going to have a different density, where a higher dense solution is at bottom, a gradual decrease in the density. So when this solution will undergoes, when the centrifuge tube with the sample is going to undergoes the centrifugation, a centrifugal field is going to be applied, where there is a fixed centrifugal field. There is no changes in the centrifugal field. After the centrifugation, a sample components based on its density, they are going to be separated. A lesser density compound is going to be separated at the top, and high density compound is going to be separated off at the bottom. So from the centrifuge tube, a different density components or a different gradient, so it must be separated without disturbing of other gradient separated component. So the recovery is very important in this centrifugation and very tedious process. So for this, a recovery of a sample can be done by two methods. One is puncturing technique and other is displacement technique. In the case of puncturing technique, where that is, from the name we can found that a puncturing technique involves a puncturing of test tube. So the test tube is going to be punctured off with the needle at the bottom, where it is going to have a higher density components separated, and at the top where there is a low density components is going to be there. By without disturbing the low density component, high density component is going to be separated off by puncturing. A high density component is going to be separated off in the fraction test tubes. After the high density components is going to be collected off with the fraction test tubes, then another test tube can be used for low density component collection. So a dropping medium which is going to consist of components which is of its similar cell components or biomolecule components. A peristaltic pump can also be used for sucking of the medium from the hole. It can also be clogged, so clogging can be possible, which is going to be not possible if a peristaltic pump is used. Next, a displacement technique. In the case of displacement technique, from the name itself we can identify that each cell component with its gradient is going to be displaced off by this technique. From the puncturing technique, we have does not disturb any other component, where the component is going to be separated by puncturing at the test tube bottom and separated based on manually with the tedious process. But in the case of displacement technique, where there is a cover at the outlet at the top of a gradient to avoid the splitting of medium. A hole is made. Through the hole a tube is passed to the bottom of the tube. So the recovery is going to be first high density component. A high density component is going to be separated off in this recovery. So a very dense solution, then the high density component is going to be where it's going to be a test tube, where there is a low density medium which is going to have a components of its similar density, and high density medium which is going to have a components of its similar density. A cover is made. In this a test tube, a tube is passed. Through this tube a dense solution is passed. This dense solution is going to have a density which is more than the dense solution in the tube. So when we pass this dense solution into the tube, this denser solution will be occupying this test tube bottom, and the denser solution in the test tube will move to top, and the less dense solution will move to another top. So the medium is going to be displaced off. There is another tube in the cover. So the low dense solution will reaches a tube, and this low dense solution will be separated off by or collected off by fractionally. Based on this, a displacement of medium with its component by pumping off the dense solution, a displacement technique can be used for recovery of the sample. So the dense solution is going to reach the bottom, which causes a displacement of gradient layer. So here we can see that there is a peristaltic pump which is very important for pumping of the solution, which is going to have 60%, which is more than the density of the gradient. It's going to have a density more than the gradient's density. So it's going to be displaced at the bottom of the test tube, which causes a displacement of each gradient. These displaced each gradient, that is light dense solution, will be separated off by fraction collector at first. So the least dense solution will be separated off first. Gradually a different dense solution can be separated off. This is going to be a density gradient centrifugation recovery, which is very important and tedious process, where a puncturing technique can also be used and displacement technique can also be used. Then we are going to see about the types of density gradient centrifugation. These types of density gradient centrifugation is possible based on its rate zonal centrifugation, isopropanic centrifugation. Now, types of density gradient centrifugation, from its name itself we can identify a rate zonal centrifugation. But in the case of rate zonal centrifugation, where the separation is going to be possible as these zones. A major differences in this both are its density. A gradient density is lesser than the sample density. A sample density is more than the gradient density. A gradient is going to have lesser density than any of the sample density. So, a centrifugation is going to be taken place for the low speed, because a density is less than the sample. There will be a low speed centrifugation occurs for the short time, which lead to separation of a component as a discrete zones. Discrete zones means as it's going to have a low density, a gradient is going to be at the top, a sample is going to be at the bottom. Of its similar density gradient, then the sample is going to be at the bottom. Likewise, another density gradient is at bottom, another sample is going to be at the bottom of the gradient. Based on its density of a sample, it's going to be separated, but it was separated as discrete zones below its gradient. So the major rotors can be used as swinging bucket rotors, because it's going to be have a decreased speed of RPM and decreased wall effect. Decreased wall effect is very needed because here there is only a small differences in the density of discrete zones is going to be formed of, which can be disturbed if there is a wall effect is possible, may affect the sample separation. So the major advantage in this is going to be particles differ in size can also be separated, but density based on density is not possible. So a separation of these components, cellular components, can also be possible in this method. As it was going to be separated based on size, a cellular component separation can also be possible. Even RNA DNA hybrid and ribosomal subunits can also be possible to be separated. Proteins which differ in small molecular weight can also be separated because it's a fragile process. But cellular components of like mitochondria, lysosome and peroxisome does not be separated by this method. So a different components is going to be separated in this method. This is going to be gradient. This gradient is going to have lesser density. This gradient is going to have a lesser density than the sample density. So it's going to form a discrete zones. Based on its gradient, it's going to form a here we can find the gradient at the top and its similar density component at the bottom. Likewise, a discrete zones can be formed in this. Next, we are going to see about isopycnic centrifugation. In this centrifugation, the sample is going to be differ in the density. So, the density of a gradient is is going to be used is not lesser like rate zonal. It's going to be similar of the sample density. The sample density and the density of gradient is going to be similar. Due to its similar density of gradient and the sample is going to be separated at the gradient itself. So, if the gradient is here, the sample is also separated in the gradient of similar density. Likewise, different sample is going to be separated off based on its density of a gradient as similarity. So the gradient is going to have a higher density so that the maximum sample can be separated off on the higher density. A continuous density gradient can be used here because a similar molecular weight changes can also be differ. Proteins with similar molecular weight can also be separated by this method by continuous density gradient usage. A sample is layered on this component because it's very tedious process. As the particle form these zones, but these zones are within the gradient itself. It's not going to be bottom of the gradient. A choice of rotor is going to be fixed angle rotors or vertical tube rotors. But a particle can be, it is based on the principle a sample is immobile at or near the bion density, which means a similar density region where a particle is immobile. That means a gradient density and the sample density is going to be similar. Due to this, a sample, that means a particle, is immobile. So, based on the density similarity, the separation is going to be taken place. The major application of this centrifugation technique is separation is going to be based on size and density. But a major part of separation is based on its density. The separation is possible not by size. A separation based on size, a good technique is going to be differential. In the case of density gradient, a separation is going to be based on density. So, a subcellular, intracellular organelle separation can also be possible. A nucleic acid fractionation can also be possible in this technique. So here we can see the forces applied here. When the centrifugation starts, where there is a continuous density gradient used, centrifugation starts and the separation of particle is possible because of a centrifugation field, and the particle is immobile when the density of it is reached. As from the isopycnic name, we can find that iso means same. So, when the sample is going to reach the same density gradient, it's going to be separated off in this isopycnic technique. But in the case of rate zonal, there is going to be discrete zones form after it reaches a lighter density component and higher density component. From the middle, it gets separated off as discrete zones. So here two forces applied. One is centrifugal force, which made it to separated off, and higher density, that is going to be bion density, which is going to provide a cushion for the component which is separated off. So these are the main differences between the rate zonal and isopycnic centrifugation. As we already made differences on each of the component, now we are going to see the differences of the rate zonal and isopycnic based on its density, instrumentation and gradient, how it's going to be separated off or differs. In the case of rate zonal centrifugation, it's going to use the sedimentation. It's going to use the sedimentation velocity principle only. But in the case of isopycnic centrifugation, the name itself we can say that same density components are going to be separated off. So based on the density equilibrium principle and the sedimentation equilibrium principle, it's going to be separated. That means sedimentation at the equilibrium is going to be separated, and that based on the density equilibrium, that means at the same density and same sedimentation it's going to be sedimented off. A gradient differs in it because a gradient density is lesser than the least dense sedimenting species in the sample. But in the case of isopycnic, so the gradient is going to be shallow, but in the case of isopycnic it's going to be steep because a maximum gradient density is going to be greater than that of most dense sedimenting species, or it's going to be continuous or discontinuous, but the gradient is going to be either increased or similar to the sample, but it was least. In the case of centrifugation in the rate zonal, it's going to be incomplete sedimentation at the low speed and short time, where very fragile component like DNA can be separated. Even though it is incomplete sedimentation, a fragile component without disturbance can be separated off in this rate zonal. But in the case of isopycnic, where density is going to have fluctuation in the density, where there is a higher density gradient can also be possible. So the complete sedimentation is possible because of the force applied like centrifugal field and the high density cushion force. So it's going to be taken place for high speed and long time. Even a RNA DNA hybrid and ribosomal subunits can be separated by rate zonal as it was shallow density and incomplete sedimentation. But in the case of isopycnic, a DNA, plasma, lipoprotein, lysosome, even cell components can also be separated by this, and also biomolecules can also be separated, but even it is not accurate as much as differential centrifugation in the case of cell components. But in the case of rate zonal, a mitochondria, peroxisome and lysosome separation is not possible as it was incomplete sedimentation, which does not provide a high speed and high time. Next we are